Imagine for a moment that you are going for an early morning walk through the jungle and you are listening to a long chorus like this. This dawn chorus can be consisting of the penetrating buzz of cigarettes and the alarm calls of lemurs and the croaking of frogs and also the beautiful melodies of songbirds from the earth to the treetop. So we can say basically all these organisms are communicating with each other both intraspecifically and interspecifically. And we can also say that the organisms which are communicating, they vocalize more during their breeding season, and it's very important and interesting to study the vocalization during the breeding season itself. And the ability of the organisms to communicate efficiently plays a very critical role in their survival and in their daily life. And with this background, I would like to introduce my dissertation titled Study of Some Aspects of Breeding Ecology and Vocalization of Central Prinia. So, sound an important mode of communication in birds. So, birds also rely upon visual and acoustic communication. Among this, they rely mostly on acoustic communication because acoustic signals travel longer distances and they can penetrate through the thick vegetation, and also they can carry a large amount of information rapidly and efficiently over longer distances. And especially for the birds which are dwelling in very dark and dense habitats where the center and recipients are not visible to each other, they communicate through acoustic signals only. So broadly, this avian vocalization is divided into two categories, that is songs and calls. So songs are comparatively complex than longer in duration and are produced by male birds during the breeding season. And on the other hand, calls are short duration and produced by both male and female, and they are produced throughout the year. So for giving a better understanding, I can give you an example of blue whistling thrush, which is a winter migrant in our campus also. So let's listen to the song of blue whistling thrush first. So this is the song of blue whistling thrush, and let's listen to the call. So from these vocalizations, I hope everyone understood what is the difference between song and call. And now the song diversity. So most of the oceans are having, or the songbirds are having more than one version of each song, and this version can be referred to as song types. And these song types consist of song units which can vary from 1 to thousands. And I can give an example of black-chinned warbler, which is having only one song unit, and let's listen to the song of black-chinned warbler. So from in this song there is only one song unit present, and we can clearly hear that one. And now I can give an example of orange-headed ground thrush, which is having multiple song units in its song. There is another interesting aspect of bird vocalization, that is singing mode. So another important interesting aspect of bird vocalization is the development of singing modes in several group of songbirds. Singing modes are the collection of song types or the repertoire of song types. So the singing modes can be explained with the help of two examples. That is with the example of Lamantamru Wabbler and also Hume's Wabbler. Let's listen to the two singing modes of Hume's Wabbler first. This is the first singing mode. This is the second one. So let's listen to the two singing modes of Dhamma. This is the first singing mode. This is the second. So both there are two different singing modes or two distinct singing modes. These singing modes differ in their motivation and function also. And there is not much known about the function and the function of dual singing modes about the wabblers and one particular species which I have chosen as my study species. And earlier only one study from India which was done by Sharat Apte in 2005 was based on the vocalization and the breeding of jungle prinia. So my study species is jungle prinia and it's having two distinct singing modes. And let's listen to the singing modes. The first singing mode is, and the second one. So these both singing modes differ in frequency, modulation, in amplitude, everything. And why jungle prinia? Why I chose this species? So jungle prinia is a small passerine bird which belongs to the family Cystibolidae, and the distribution of this species is throughout in India and except in northeast and Himalayas. And the reason why I chose this species is this is a species under the Cystibolidae which is very poorly studied and very less known about its breeding and growth. So, and the function of two singing modes needs to be studied in future also. So I chose this species because of all these reasons. Here comes the objective of the study. I have mainly two objectives. The first one is determine the difference in singing modes during different breeding stages of jungle prinia. And there were two questions. Is there a sex-specific function for the two singing modes? The second question was, is there any change in the use of singing modes with time? And the second objective was to investigate the influence of habitat in the song duality of jungle prinia. And the question was, does the habitat is having any influence on the two singing modes of jungle prinia? And the study area. So I chose the XVII Community Reserve in Mohali district of Punjab as my study area. This community reserve is a part of Shivalik Hills and is located in the boundary of three states, that is Punjab, Haryana and Himachal Pradesh, and this study area spreads around 13 square kilometer, and the intensive study area which I was focusing on was around 2.3 square kilometer in the XVII Community Reserve, and the area was predominantly dominated by the forest type, that is tropical dry deciduous forest, and the abundant thorny species also were present there. And 6 sampling sites were selected after the preliminary survey on the basis of the presence of single prinia. Here come the methods. Field work was carried out from early April to early June, and I quantified the following singing behavior, vegetation, bird density, and the song function and transmission in the 6 sampling sites I have selected. And let's get into the details of the methods. So, singing behavior sampling and point transect. So, singing behavior sampling and point transect were conducted in 100-meter radius, and I was standing in the central point and I was recording the singing mode, the time, and the bird location. And the point transects also were conducted in the 100-meter radius. And I was observing the movement and also the response between the birds also to ascertain the individuality and location of the birds which are present inside the 100-meter radius. So the vegetation sampling. In vegetation sampling, I laid 4 plots of 10-meter radius in all cardinal directions, and in 10-meter I collected the parameters related to trees, and in 5-meter I collected the shrub parameters, and also in 1 meter I collected the percentage of grass cover, leaf litter, and the bare earth with ocular estimation. And here comes the playback experiment. So playback experiment was done using a speaker and a smartphone. So the playback experiment was on the basis of this protocol. I divided the response of the birds on the basis of their behavior into 4 categories, that is strong, moderate, mild, and no response. And the scores were assigned respectively. The strong response was given 3, and moderate response was given 2, mild 1, and no response 0. And these are the 2 typical responses we were observing while doing the playback experiment, and these behaviors were categorized as the strong response towards the playback experiment. So first one is the nose diving. The bird will sit in a high perch and they will be continuously doing this nose dives while doing a playback experiment. So the second one is the direct movement towards the speaker. So then we conducted the transmission experiment. So in this we played the singing mode A and B, and we recorded it at a distance of 20 meters away from the initial point. So we can clearly see from this one there is a single difference in the amplitude of both singing modes. Here we can see the amplitude of the B sound is higher than the A sound. So it means that the A sound and the B sound is traveling more than the A sound. So let's go to the analytical method. So use distance software for the density estimation of the male bird, and the linear regression was used to find out the relation between the habitat parameters and the differential use of singing modes. And the differential use of singing mode was calculated using this formula. The number of boards of the singing mode by sum of boards of all singing modes. And the percentage cover and the average of all the percentage parameters which I have calculated or recorded clearly was calculated using this formula. And the study period, the whole study period was divided into 3 stages. So the pre-university phase, so it was divided into 3 stages: early, mid and late. And here comes the results and discussion. So this is the main song playback responses for singing mode A and singing mode B with the singer. So here we can clearly see that the response of birds towards the song A playback was very strong when compared with the song B. So from the next graph we can see here the response singing mode, we can see the response towards song A in the first graph and the response towards the song B. So that means the response towards the playback experiment done with song A and the response towards the playback experiment done with song B. So we can say that I conducted a number of playback experiments with using singing mode A, that is 22 times, and in every playback experiment I found response. We have observed the response. Then in the response of song B, singing mode B, we conducted almost 26 experiments and 19 times there was response and 7 times there was no response at all. And from this graph, this is a diagram, we can say both the graph can give us the idea of the response of the bird for both singing mode was in singing mode A only. They didn't even respond in singing mode B anytime. This graph gives the differential use of singing modes across the different sampling sites. So from this graph we can say the singing mode A was dominant across all sampling sites. And then we compared the differential use of singing modes with the different stages of play. So we can say from this graph, there was a clear decline in the singing mode B, usage of singing mode B as the radiation progressed, and the singing mode A was increasing. So I used linear regression and I regressed all the habitat parameters with the differential samples. So only one positive relation I could find, that is the relation between differential use of singing mode B and the subcover across the different sampling points. So wherever the subcover was more, the percentage subcover was more, the differential use of singing mode B was more. So I found it significant. Then the differential use of song B versus main bird density. Here, unfortunately, as expected, we couldn't find any significant relation, but comparatively, we found a very weak relation. So, conclusion, I can say, the singing mode A was dominant across the stages of pre-nesting phase, and singing mode A is used for aggressive main-main interaction. So we concluded from the playback responses, that was earlier I have mentioned, all the playback responses was in singing mode A, and as the breeding stage progressed, the singing mode A only was increasing. From these 2 points we can say that the singing mode A's function was to communicate with male or male-male interaction, aggressive male-male interaction was the function of the singing mode A. And the singing mode B is used for long-range communication. So from the transmission experiment, we could find that the B song was traveling more than singing mode A. And also from the relation of percentage of cover and singing mode usage, there was a positive relation between the sub cover and the communication. So wherever the sub cover was more, these birds were using the singing mode B so that they can communicate longer distances. And also the response to playbacks of singing mode B, and from this we concluded that the singing mode B is used for long range communication. And also the influence of singing mode with habitat, as I have mentioned earlier, there was a relation between the shrub cover, which was the shrub cover percentage shrub cover was consisting of mainly grass species which was considered, which were categorized into the shrub cover, that is Nerodia and Nature, because of the comparatively bigger, larger height and the presence of thicker cassets. So, way forward. So, if we could have initiated the sampling well before the onset of rainy season, we could have find the clear-cut functions of both singing modes. And if we would have taken the continuous recording using automated recordings or audio modes, we could have got a larger sample size, and we could find the difference in singing modes along the day. There is a change in the singing pattern. And also color banding and individuals color banding also could have been a very good attempt to identify the each individuals and to assert the location and individuality.
Thanks, Vignesh. Questions for Vignesh? Hello, Vignesh, can you hear me?
Yes, sir.
Yeah. It was a very interesting talk and a very interesting presentation. A couple of questions. The first question is, in terms of the responses, variable responses to the two singing modes, did you see other studies also which show this sort of variable response with similar species or with the same species?
Yes, sir. So, in earlier studies and all, a lot of studies have been done earlier in automating warblers. So, especially in the species belonging to the genera Vermiola, Citoparangol. So, in that, they have found a similar kind of interaction with the first singing mode, which is found to be very aggressively responded by the individuals. It was taken as territoriality, or the function was taken as territoriality and male-male interaction, like what I have found here. And the singing mode B was mostly like to communicate with the females, but I couldn't find any function like that.
Okay. Yeah, so the next question is in terms of looking at, can you hear me?
Yes, sir.
Yeah. So in terms of looking at your variation in singing modes, when you assume that male-male aggression is the prime reason for your data showing more responses to singing mode A, did you find, sort of account for the differences in male, densities there, or similar bird densities there, or with other birds?
Yes, sir. So,
Could there be, the question is basically, could that also contribute to the kind of responses that you get? For example, because if there are more number of males in a habitat that is more conducive, you would have more responses. If the habitat is less conducive, you would have less number of individuals. Maybe the responses would vary slightly if there is any variation of such.
yes, sir. Unfortunately, the image we selected was almost uniform. So, we didn't find any relation with the density. Earlier we were expecting that if the density is more, there will be more interaction between males, competition between males. So there will be an increase in the singing mode A gradually, but there was no.
And what about the, sort of ratios of males to female ratios? Those could those also have?
But I can say, sir, like
because even if it is there, that this is also a relative context. Because if there are more females, in that area, or if there are more males, that ratio is different. Do you think that also would affect your sort of data?
but the females were comparatively very aggressive and they were not coming out several times. Like in the mid study period only they started at least coming out, the pairing and all started. And I can say that